Hello, everybody, and welcome. Thank you for tuning in again. This is part 2 in the Mydecine Speaker Series here. Today, we are going to be covering microdosing. It's been fun for me to promote this one because Microdose is actually finally starting to do some microdosing content. So we want to thank Mydecine for coming on. We've got some fantastic speakers. I'm not going to be monitoring. We have Elie monitoring, and I'll pass the mic over and let you introduce all the folks from Mydecine here.
Great. Yeah, well, thank you. And it should make for a good conversation today. Obviously, microdosing, psychedelics, I feel like it's just kind of arrived on the scene as this thing that's entered the public consciousness and is something maybe that tech bros in Silicon Valley are doing as some sort of life hack. But there's some real science and medicine behind the approach with potential for treating all sorts of different diseases such as depression and other mood disorders. And so to discuss that potential, I'm joined by Dr. David Erritzoe, a psychiatrist and brain imaging specialist from Imperial College London in the UK. Dr. Vince Polito, a cognitive neuroscientist from Macquarie University in Sydney, Australia. And Rob Roscow, Chief Scientific Officer of Mydecine, a mycology-focused biotech company in Denver, Colorado. And I'm science journalist Elie Dolgin, and I'll be today's moderator. Hi, guys.
Hi.
So before we get into the topic at hand, I thought it would be good to start with a little bit of some historical context. And as I understand it, Albert Hofmann, the Swiss chemist who actually discovered LSD and psilocybin, he had actually discussed this idea of microdosing and using small doses and that it could have therapeutic value all the way back in the 70s or so. So I'm wondering, Dr. Polito, or Vince, if we're going to use first name, sorry, could you just quickly give us a rundown of what was known about microdosing kind of from the early heyday of psychedelics before LSD and magic mushrooms and other substances became illegal, and kind of there was this big pause essentially hit on the research agenda? Oh, Vince, did you hear my question?
I'm sorry, I cut out briefly for about 20 seconds.
No. Sorry, I was just wondering if you could, just to set the stage before we get to what's understood today, what, in terms of microdosing as something different than macrodosing, what was known back during the early heyday, the kind of counterculture research days of the 50s, 60s, and 70s?
Yeah, interesting question. So microdosing, as we think about it today, does seem to be a relatively modern phenomenon, although there are some hints of people experimenting with things similar to microdosing in the past. So I didn't completely hear your introduction, but I think you mentioned Albert Hofmann talking in an interview about taking very small doses. That was kind of something that appeared in one published conversation with him that there doesn't really seem to be much other evidence of. In the 50s and 60s, it doesn't seem like there were too many people that were taking very small doses, although in some of the early experiments that took place in those days, there were some studies that had very low doses as a control condition in studies looking at high dose psychedelics for other purposes. And so when you go back and look at some of those studies, they sort of do fit in with what we would consider microdosing today. And so there are some hints, even back in that early research, that very low doses were having some effects, although it wasn't very well understood what was going on at that stage. If you go forward then into the late 70s and early 80s, there are some hints also there of people starting to take small doses. There was a group in Berkeley that does seem to be experimenting with very low doses of LSD in the early 80s, but it didn't really catch on in popularity until probably the last 5 or 6 years, popularized a lot recently by Jim Fadiman and his work in the Psychedelic Explorer's Guide. And then, yeah, as you know, there's been this kind of subsequent really intense interest in popular culture in microdosing and the possibilities that it can lead to.
Yeah, and we'll get to all that for sure. But just again, to sort of paint the picture of where we are. So more recently, research on psychedelics has kind of come back into fashion within medicine and neuroscience. But most of the research has been on macrodosing with large doses of these hallucinogenic compounds. And David, I think you got into this field doing some of the basic neuroscience brain imaging studies on users of ecstasy, magic mushrooms, other drugs. So maybe you can just quickly tell us where the field stands in terms of the therapeutic benefits of full dose psychedelics.
Yeah, thank you. In terms of full dose, a lot has happened in the last decade, and it has actually, I would say, happened even quicker than I think we thought in our team. So we have been positively surprised how much is it taken on. But we conducted some full dose psilocybin trials recently. The second one in depression that will soon come out, and during that trial. While we were doing that, a couple of companies have launched multi-center trials across a lot of countries and have raised funds and investments. Some of it is non-profit, the idea, and some of it is pro-profit. And they are now kicking off and have started recruiting on different sites, both in Europe and in the US, in order to treat depression with full dose psilocybin. So I guess that is the thing within psychedelics that is furthest, if we do not refer to MDMA as a psychedelic, which we typically don't, but the model obviously overlaps. MDMA is further ahead, but the psilocybin for depression is really happening. So within a few years, there should be conclusive results whether it will come out as a licensed treatment or not.
Okay. And just for people who aren't familiar with kind of the clinical trial process, you start with phase 1 for safety, phase 2 to show that there's kind of a sign of efficacy, and then you do a larger phase 3 to show that for sure. And so I guess, so it sounds like whether it's general depression or maybe people are familiar with, people with cancer who are trying to deal with the depression associated with their terminal illness. I mean, this research is basically at that confirmatory stage. Is that right?
Yeah, absolutely. And the trial, the large trial that is furthest ahead, is targeting treatment-resistant depression. So patients who haven't responded sufficiently to conventional treatments, they are the ones going into one of the trials, the one run by Compass, and then other people, Zona, and soon maybe also others are running it in major depression, so just people with depression. And you're right that Johns Hopkins was absolutely world-leading place for psychedelic science. What was it? Sorry, I lost the thread. Sorry.
Right, but I think with the cancer patients and stuff.
Yeah, exactly. The cancer, absolutely. End of life. They had done that. That was their early studies and very solid studies, and all of them have shown very positive results. And so it's not phase 3 done first. It's 2B that will then go over into phase 3 for most of these trials. And then they're also being planned similar work, a step backwards to do phase 1 and 2 with DMT as well. We are setting that up as well. So that's also going to be tested for depression, which we can,
And I think MDMA is pretty far along for PTSD and stuff. And
absolutely, that's furthest ahead. So that will be the first thing to be licensed if the results hold.
Yeah. Okay. And last question here to set the groundwork, I promise, and then we'll get into the actual science of microdosing. But Vince hinted at this, and I'm wondering, Rob, if you can elaborate on. So Jim Hanman, about 10 years ago, comes out with this very influential book, The Psychedelic Explorer's Guide. And that kind of puts microdosing on the map. I mean, maybe not for researchers, but definitely in the, among the curious public. And so maybe, Rob, can you explain a little bit about the importance of that book and how especially how it has influenced both perceptions of microdosing in the popular culture, but also especially within kind of the medical establishment and at companies like yours, and maybe even helping to launch Mydecine?
Absolutely. So I think you're hitting the nail right on the head, that publication is really kind of a breaking point where people are seeing this as potential, and then there's a lot more instances of other published books, other popular cultural references. So it kind of snowballs from there as far as public perception, interest, and then people going out and experimenting on self-medicating this way themselves. And so as a researcher looking at this, what's really exciting is there's enough self-reporting that there's definitely something at play here. We're seeing a strong signal from the general public that when they do this themselves, without really, medical safety rails, if you will, that there's, at least a benefit in people that are reporting this. And so what we see is this is a, really strong evidence of potential, and the need to understand what's actually being, the mechanism that's at play and what's actually medically going on, so that we can better describe when and if and where this is really appropriate for patients. And so that's the exciting thing with what you're describing there, that because the public has become so interested, it's provided an opportunity to see this value, which then medical, research, clinical studies can follow up with, and we can really understand, the nuts and bolts of what's going on, and provide access to the best access to the most number of patients, which is really the ultimate goal.
Okay. And just so we're all on the same page and talking the same language here, Rob, can you, I probably should have done this right off the bat. When we say microdosing, and there are some differences in how people use the term, but for the sake of our conversation, at least, and in general, as it's widely used, can you just give us kind of a broadly accepted definition?
Absolutely. So the general perception is, and the general sort of protocol that people would be doing here is trying to take about 1/10 of a dose that would be the normal full hallucinatory experience. So that's the kind of goal point that people are using for their dosing. And beyond that, people are really looking at the time periods that they use them, and you'll see that in the practices that people do, they have specific every couple days, this sort of thing. But what we're really talking about is a supplement sort of use of these compounds at a very much sub-perceptual level. The goal is to not have any of the hallucinatory effects, but have a mood lift or a cognitive boost of some sort. So,
okay, all right. Background out of the way. Let's actually talk about the topic at hand. So, David, you and your colleagues, about a year and a half ago or so, you published this. It's a very useful review article that people might want to check out. It was in the Journal of Psychopharmacology, and where you did what is called kind of a meta-analysis or a systematic review of all the literature to really get at what was the state of knowledge, at least in the academic domain, around microdosing. And so I'm wondering if you can just quickly tell us a bit about the kinds of conclusions that you were able to draw, realizing that was, 18 months is a long time in the state of this science, which is moving very fast. But maybe you can just set the groundwork for where things were, at least last year.
Basically, the conclusion was that there's a lot of anecdotal evidence, not a lot of safety evidence. One of the things raised in the paper, by the way, the first author is Kip Kuipers, who's a professor in Maastricht, so not without him. So it was a sort of a collaborative paper. But the conclusion is very anecdotal. Safety-wise, we brought forward the 2B receptor as something to think about, because it's expressed in the heart valves, and previous medications have been taken off the market because they induced fibrosis in the heart valves, and that was mediated through the 2B receptor.
This is the serotonin receptor.
Serotonin receptor. Yeah, so the psychedelics work in that system, in the serotonin system in the brain, but they obviously will also be circulating in the blood system, and will hit targets for where they also would hit in the brain. And some of those receptors are in the serotonin system are sitting more outside the brain, and including the 2B. The psychedelics, they do not only target the main sort of key psychedelic action receptor, which is called the 2A receptor in the brain. It's serotonin 2A receptor, but also this 2B receptor. And there's no reason to believe that sporadic full doses haven't been cases with any problems with the heart. But if people suddenly start taking a bit like if they take SSRIs often for many, many years. So if you kept on microdosing forever, could that potentially affect the heart valve? So maybe some animal experiments might be a thing to do at some point. Maybe some people are already doing it. I could imagine actually that there would be, in order to test that, to be a little bit more on the safe side. And then basically the paper was concluding that there's a lot of promising sort of but weak evidence that it can potentially improve mood, improve anxiety, and also stimulate cognition, memory, and creativity. All these things that microdosing that are becoming more and more known to do. However, a lot of people, I think at this stage, and also even more than a year ago, thought that the few data had come out that supported were really basically concluding that psychedelic works. I guess it can be confusing with evidence because there's so many levels of evidence. One thing is the anecdotal, and where there's no placebo control, and also sometimes some of the evidence comes from retrospective. So if people are asked, you microdosed, how did it go? What improved? What went worse? That's a pretty confounded way of it. It's fine. It's better than not trying, and it can start producing some hypothesis to conduct more solid trials where they are better. They are designed in order to prove whether this is working beyond placebo levels. So I would say the conclusion was that studies need to be done, and studies are being done. And as people might know, particularly my strict group are doing quite a lot of sort of very solid microdosing work where they're placebo and different doses and so on. Yeah.
Okay, I will get into some of the placebo-controlled studies, but I also just want to get Vince here to talk about the study that you published last year in PLOS One, where not placebo-controlled, but pretty rigorous, where you tracked the experiences of nearly 100 microdosers for, I think, about a month and a half or so. I'm curious, how did your results from that square with what David just said about the general state of the field?
Sure. So this study that we ran was a fairly broad study. We had quite a number of different measures that we were investigating with microdoses, and we were looking both at the sort of immediate daily changes on days that people microdosed, and then also longer term changes. We tracked people for a period of about 6 weeks, and we wanted to see if there was any change from baseline to the end of the study on quite a battery of different psychological and cognitive measures. This study was published in 2019 actually, and we collected data for it in 2017 and 2018, so a few years ago now. This was, I guess, the early stages of research with microdosing, and one of the main sort of motivations for this study was to try and see if we could work out what was going on with the reported effects that people were claiming about microdosing in terms of their beliefs and expectations. So as well as tracking what happened with microdoses, we also asked people about their expectations of what they thought microdosing would lead to. And this was sort of, it wasn't as rigorous as having a proper placebo control, but it was our way of trying to identify the role of expectation in some way. And so we found, firstly, that there were some quite interesting changes reported amongst the people who microdosed. They reported reductions in depression, reductions in stress, reduced mind wandering, and increase in something called absorption, which is like someone's internal attentive capacities. And we also found a small increase in neuroticism, which was a little bit surprising and a little bit inconsistent with some of the high-dose psychedelic research. So it's quite a curious combination of different effects that we found. But perhaps even more interesting was that we looked, as I said, at people's expectations and tried to see how these findings squared with what people were expecting to happen. And we certainly found that people had very strong expectations about what microdosing would do. People basically thought that microdosing was going to increase all of the positive psychological variables and decrease all of the negative psychological variables. And so in some ways, it does seem like what people believe could be having quite a strong influence on what they then report. But interestingly, we found that the things that people most expected were not what the microdoses actually reported. So, for example, people most expected increases in creativity, increases in mindfulness, and they weren't actually effects that we found in our study. The people didn't really have strong expectations around the absorption or mind wandering that we did find, and so that made me, reasonably optimistic about the possibility that microdosing is having some kind of pharmacological effect here, or some effect beyond placebo. So I think there's definitely expectations and placebo-like effects at play, and I think some of David's more recent research really speaks to that more clearly. But it does seem like there are potentially perhaps some subtle changes that microdosing is influencing that could be of interest and worth pursuing further.
Yeah, I guess that's important because, as you guys are both saying, I mean, these were typically, open label or, self-prescribed. These are not placebo-controlled studies, and so there's a lot of opportunity for the placebo effect. And the placebo effect doesn't actually have to involve a pill. It's just this idea that if you think something's going to work and have an effect, it just well might. And it makes me think, it reminds me a little bit about the whole CBD craze, where people are taking CBD under the tongue or smoking hemp flower, and they're expecting it to kind of be the cure-all for everything. And there's anecdotal reports of that. But if you actually look at the science, I mean, there's definitely value to it. And definitely for kids with epilepsy is probably the only thing where it's gone through the full traditional medical rigorous process of proving a medical value. And there are definitely hints of other things. But what more data do we have to show that this isn't just one big reporting bias and a lot of people getting out of microdosing what they expect to put in, And either of you guys want to take that one?
I guess it's, I can come with my little input here. I would say that it's easy to answer because there is really not much, there isn't any evidence, not for continued microdosing over a sustained period, because the trials haven't been conducted yet. And then you say, why has that not been conducted when there's all this hype? It is because it's a relatively new hype, and therefore the field, have been firstly focusing on the full dose because there was a whole backstory and good reasons to open that line of research and try to bring them into different avenues for therapy. But in terms of microdosing, one of the reasons I think why it has taken a bit of a time before people are setting up the big trials is that it's extremely expensive, and the convincing people with almost no evidence from the start is obviously difficult, and they're also not very practical to run such a study because we're speaking about classified scheduled compounds, and that means that one thing is doing a full dose when you invite people into the lab on a few occasions, and one of them or two of them with a full dose psychedelic experience. That's one thing, but if you have to give people, 20 doses over a period of time, or over a month or two, they still would need to come in a lot of times because you can't send people home with an illicit drug. And that also means that if you work in Google and you want to participate in such a trial to see if you stimulate your creativity, then in a way you'll be stranded in a lab several times a week instead of going to work. It'll be taken out of the naturalistic context, unless you could be given the approval to basically give people doses to take home. And I think in Auckland they might be able to do that. It looks like they will be doing something like that, which is really great in New Zealand. Maybe not all the doses for many weeks, but maybe for a week at a time or something like that, so that it doesn't, even if you took all of them, it wouldn't be, I mean, it's not dangerous probably to take quite a lot of microdoses if it's only 5 to 10% of a full dose. But anyway, that's what might be happening in the future. So the trials will come, and you can also conduct it in a lab. It's just logistically, cost-wise and so on, quite impractical and difficult. But yeah, so I think we can't conclude anything at all whether it works in over time microdosing.
Well, let's talk about some of those placebo-controlled trials first, perhaps with the ones that you guys are running. I will come back to you, David, on how the clever way that you guys have come up with a way to run a placebo-controlled trial from people's homes. But you mentioned that one can do it from a lab, which I think is what Vince is now planning with Rob and stuff. So can you guys talk about? I'm not sure what the status of that study is, but obviously can you talk about the methods and the goals and any, I don't know if there's any results, or you can give us an update on recruitment or anything.
Sure, I can tell you a bit about our upcoming study. We haven't started recruiting yet, so I can't tell you anything at all about results or anything that we're finding. We'll be recruiting shortly. But what we're trying to do in this study is look at really the immediate effects of microdosing. So this time, not so much focused on changes that result from a long period of microdosing, but really we're trying to see if we can find any cognitive or physiological markers of something like a microdosing state or state-based changes related to microdosing. So we're going to get people who are already microdosing, regular microdoses, to come into the lab. We're not going to administer substances or provide substances. We're just going to ask them to come into the lab on a day that they're microdosing and a day that they're not microdosing. We're going to run a series of brain imaging tasks using MEG. The main sort of aim of that part of the study is to look for changes in resting state and also changes in novelty perception or pattern recognition. That's the sorts of tasks that we're running there. And we've got some other behavioral and cognitive tasks on a similar theme, really looking at whether people's low-level attentional responses to stimuli are different on the microdosing compared to not microdosing days. And as well as that, we're going to look at a series of physiological biomarkers to see if there are any sort of more easily definable objective indicators of what happens when someone microdoses.
What kinds of biomarkers are you looking at?
So we're looking at things such as BDNF, cortisol. We've really got quite a suite of different genetic sort of biomarkers. Maybe Rob could speak a bit more about the rationale behind some of those measures.
Yeah, I'm happy to jump in there. And so the goal really is as was sort of already highlighted by David and Vince, is that we're seeing these hints at stress relief, depression reduction, mood lift, etc. And these are things that have known biomarkers already with them. And so the idea is that if there's a less of a signal to see in the outward perception, because the whole idea here is that this is a lowly perceptible or not perceptible treatment that then is providing benefit. So really those sorts of biomarkers provide better visibility into what we're actually looking at. And so that's the interest there, is it gives us a better tool to look at this situation.
And you're recruiting from people who are already microdosing, or also trying to find people who have treatment-resistant depression and maybe are just curious to try it or something?
No, this study isn't really looking at clinical effects of microdosing. We're recruiting only healthy controls for this study. A couple of reasons for that, but the main reason is really that is a much easier sort of study to get approved by ethics. I mean, any of this kind of research at this stage is it's a little bit contentious, and there are certainly some interesting back and forwards with ethics committees to make sure that we do everything in a safe way that's really going to care for participants. And so at this early stage, when there is so much that we don't yet know about these kind of immediate state-based effects of microdosing, we're aiming to try and look at these measures in a healthy population, and perhaps down the track move on to seeing if this is more appropriate for people with depression or other mental illness.
Made this a bit of an aside, but how much of a confounding issue is that in our interpretation of the data, that primarily these studies are happening on people who have chosen themselves to microdose, presumably don't have a diagnosed illness. I'm not sure if that's going to be an exclusion criteria in some of these to get by your ethics committees, and that these effects that are being measured on mood and other things that might track with improvements in major depression or something that ultimately is presumably the goal here is to find uses for these treatments in serious mental illness, but we're not actually, by and large, studying this treatment in that population. So how much can we really extrapolate, and how much is that put a giant asterisk on kind of almost all of this research?
Yeah, I think it is a concern, and something that's certainly worth us thinking about. I guess the way that I sort of feel about it is that, we've got some evidence, for example, from the earlier study that I told you about, that was also with healthy participants. And even though those participants didn't have any clinical levels of depression, anxiety, stress, we did still find reductions in those measures amongst that population. So I guess the rationale for doing this work in this way is that we think about something like depression as more of a dimensional characteristic or property. It's not the case that people who are healthy have absolutely no depression or properties of these kinds of psychological conditions. And so if we can see some changes amongst this population, then that could be an indication that it is going to have an even stronger effect in clinical populations. But yeah, you do raise an important point. We don't know for sure if that's how microdosing reacts in these different samples. It's possible that people with these different clinical conditions are qualitatively very different from the healthy controls and might respond differently. So that's certainly an understudied area and something that is worthy of more attention. I do agree with that.
And the other thing that kind of raises a red flag in my brain a little bit is, so we've been talking about microdosing as if it's like a clearly defined thing, and Rob obviously gave us a working definition before, but presumably most of these subjects are procuring their own drugs from the street. They could be, maybe you're differentiating between LSD and psilocybin, but we could talk a little bit about, obviously these are different drugs. They work through similar receptors, but have different effects. And I don't even know, is everyone using the same microdosing schedule, the kind of, the one that was laid out in the Fadiman book of kind of 1 day on, 2 day off, or presumably everyone, that you're going to have a lot of variability in that. So I just wonder, and maybe Rob, you can fill in here, because obviously Mydecine is trying to standardize some of this stuff to some extent, but that just, again, it just seems like it's going to put so much noise into the data, and so it's almost no wonder that we know so little, because we're just like, these aren't, I appreciate that because of regulatory, ethical, legal restraints, this is almost the best you can do, but it does seem like a huge limitation in our ability to really get at good, solid scientific answers about what.
Yeah, you're exactly right that it is a noisy research situation right now. And the thing that's really promising is that you do have people that are continually self-reporting on these really positive anecdotal benefits. And you see that across multiple geographic populations. And, now we're talking about this has been popular for about 8 or 9 years. And so you're seeing this across multiple groups that are separate. That's actually in a population scale, a really strong signal. But the research that's then tried to, at the first instance, like David and Vince have just described, it's not catching the full breadth of that value. And so there really is an undercurrent, most likely, of, people self-medicating for treated conditions, for instance. And so that's where, we as a company see a lot of potential. But, Vince very, very nicely outlined, and David as well, the reason as to why you can't just directly go to a, disease population with the microdosing. And so I think really the ability to look at minute effects in patients that are maybe, considered in a study context as, normal and healthy, but could still have minor stress, minor anxiety, minor depression issues. I think that really is the stepping stone way of moving into that. And, the summation of the macrodosing work that's going on and really moving forward nicely, plus then understanding, these microdosing results will give us a better mechanistic understanding of what's going on, and that will kind of help illuminate everything that we're talking about. But it's exactly right that it is a messy situation with the data right now, but a lot of promise.
Can I say something? In a way, I would almost turn it around and say, I think, yes, that's right. But in a way, it's a privileged mess, because if we think about it, this is just a bonus that we have something that is being done by real people out in the real world that we can then study in order to gather evidence that then can form the base for proper trials. The real trials will definitely come, but all this that we can sample interesting evidence from the public just actually speeds up the whole thing to some degree. But you have to start somewhere, and for a new thing like this, it would have to be such surveys in, I guess, opportunistic surveys where people who are just, on their own initiative planning will participate. So I think it's a very privileged sort of field, in the sense that we can collect a lot of interesting data from that, which is not, conventional.
Well, can you talk a little bit, David, about your self-blinding microdose study? Because I think that's it's a pretty clever way of, as you say, taking advantage of the fact that this is happening, but moving beyond. And it's going to involve self-reporting, but you're also kind of creating a way that people who are doing this and are interested in furthering the research agenda can create more robust data through a blinded, placebo-controlled fashion. So tell us a little bit about that study.
Yeah, maybe before I mention that study, I just want to say that we have done 2 survey studies, and I think one was actually, the first one was published today by Laura Kartner.
Today?
Yeah, I think she just wrote an email that it was had been accepted, so it's going to come out from our team, and that's not that is not a blinded study. That's a survey like the others. So you can obviously collect data retrospectively, where you just collect information about what people got out of microdosing by a lot of questionnaires. Then you can have prospective, where you follow people from before, during, and after the microdosing period, which is of course stronger. And this first study that we did was one of those, so a survey study, and it showed increases in well-being, and a lot of other interesting parameters, giving optimism about microdosing working, same as Vince and other people out there studies. But we on the side of that study, we also developed this self-blinding method. It was developed by a collaborator who now works for our team, Bala Shigeti, and it's a very smart idea, where he created this sort of manual, so we could do sort of citizen-based science, where people out in the world who were planning to microdose, they consented online to almost play this game, where they would allow themselves to potentially be under placebo condition. So people needed to acquire different things, some small bags, print out some labels, some QR codes, some envelopes they need to get, and then not least some capsules that was not transparent, and then they put their microdoses into capsules, and then they put them into small bags with labels. They put them into envelopes and mix them in a certain way, and then a third of the participants, based on the way the manual was made, the algorithm behind it, then a third would end up having taken the planned microdoses that they were planning from the beginning before signing up to the study for 4 weeks, and then another third would end up with 1 week microdosing, 1 week placebo, 1 week microdosing, 1 week placebo, and the last group placebo all 4 weeks. And then we collected a lot of baseline data. Remember, it was sort of pseudo-normalized study online, so the only thing we kind of needed was for contact with the people with the email address. We didn't need any personal information, as you don't do in surveys, but then online they were able to fill out tons of questionnaires, solving tasks during the course of the study. And then in the end of each of the 4 weeks, they could scan in their QR code from the envelope that they had been using that week, and then we could see whether it was an envelope with microdoses or placebos. So therefore we knew what they were taking, but they didn't, unless they sort of opened the other envelopes. That you produced 8 envelopes, and you only need 4. So there'll be 4 envelopes that you put aside. So you could kind of break the blind pretty easily. You could also open the capsules, obviously. Yeah. So with all its weakness, which is that we can't control what drugs they're using, we can't control the dose. So if people got it completely wrong or had an LSD blotter lying in the window in sunshine for 10 years, then which probably would remove a lot of the LSD, then obviously that could be problematic. So in these kind of studies, it's massively it's an advantage to be able to measure dose, and hopefully we will in our next trial, but we couldn't in this one. And also we obviously can't check whether people are cheating, but we have no impression that people do. On the contrary, we had people, because when they had finished the trial, they could be informed whether they were taking microdosing or whether it was placebo. And some of the people who were curious and got the answers and some of the results, they didn't believe it until we then wrote back to them, go in maybe and open the other envelopes, and you can see your real microdoses you haven't used yet. You basically had placebo, because we had people explaining that it was the most profound thing, and it had markedly better results than any treatments before for depression. This wasn't a depression study. It was again opportunistic sample, so it was people who were healthy, but it was also people who were trying to self-medicate, and we acquired all the information about for what reason, and we also had baseline and follow-up measures of depressive symptoms and so on, even though if a lot of them score almost 0, it's not very informative, but there will be people who score above a certain threshold on a depression scale. And
sure.
Yeah.
But so it sounds like some people definitely had a placebo response, but on the whole, have you done early analysis, any data you can tell us about?
Yeah, we have basically completed the paper, and that paper, now I said the other study comes before, but this one has also just been approved for publication, so it will come very, very soon in the journal eLife. Yeah, we have the full data analyzed, and it basically shows that if you ignore the placebo group and just look at the microdosing, which would be the same as a prospective survey, it basically confirms all the previous surveys. It looks amazing no matter what you look at. Almost it improves. It helps you with anxiety, depression symptoms. It increases well-being, which is the main outcome. Typically, well-being is the main outcome when it's not a clinical population per se. So it basically leads to multiple benefits. However, when we then take into account the placebo, which we had in this study, then it doesn't separate from placebo. And it was actually quite.
What was your sample size?
Yeah, that was what I was going to say. It's not a small sample size. It's 200 people. There were 240 doing the baseline, and then just under 200 completing the 4-week measures. Then their follow-up after 1 month So it was 4-week regime with whatever number people wanted to of doses they wanted to use. Most followed the example that I think we took from Fadiman. So actually most people did it very similarly, maybe different days during the week, but there were rules. They had to microdose on a Thursday and they could never microdose on a Sunday, so that we could collect acute measures on a day where we knew they had taken it that morning, and then on Sundays we could sort of think about it as a subacute measure of the previous week. And then in the end we had the accumulative data collection after the 4 weeks, and then a follow-up after 4 weeks, and then a few more people had dropped out by that time point. So in a way, I think it's the largest placebo-controlled psychedelic study in this new era of psychedelics, because no full doses are up in near that number yet. So in that regard, it is obviously very disappointing for people who have been optimistic about microdosing, that this simply doesn't look very promising. Then you can say, oh yeah, but it might work in the clinical population, and absolutely. And this is with the different Achilles heels that this study has. Real big trials should be conducted, because it would be, fantastic if it could work. So,
one thing that's an interesting point of context there is that if we think about other treatments that are available for depression, especially lower levels of depression, they also in their own trials have major issues of separating placebo effect, and are not necessarily this black and white sort of response. They're showing slight improvement versus placebo in most of the best research. And so that's an important point to remember, is that the data that we're seeing is actually quite comparable to that sort of data. And like David's saying, is that larger sample sizes may address those issues as well.
Yeah.
Yeah.
Well, my wife's a psychiatrist, and she'd be the first person to say the placebo effect is like the best thing in all of mental health care. But you do want to hopefully see separation of those curves and differentiation from placebo, especially if you want to get anything, approved as an actual medicine and prescribed by doctors.
Yeah, I guess you first need the money to run the trials, right? And then you need the approvals if the trials, fall out positively. And I agree with Rob that some of the SSRI work, and obviously those medications for depression, anxiety, and other conditions for that matter, they small effects, but they do separate. I mean, they do have positive effect, otherwise they wouldn't exist and be prescribed. So I would say there simply needs to be beyond placebo. But I guess from people, very optimistic about microdosing will probably, say that our study, but it was people's own drug, and let's do it. And I agree, it should be done, but studies have to show benefits beyond placebo, including in this field. And SSRIs, they do show that.
Well, just to finish, to complete the placebo-controlled discussion, I mean, can one of you speak quickly to especially like what the group in Maastricht has shown, or the group in Basel, or like other places that have recently reported some placebo-controlled microdosing results?
Say quickly a little bit about some of those studies. So, when I hear David describe the results of his self-blinding study there, it certainly does make me feel like, okay, wow, it seems reasonable to think that perhaps a lot of the positive findings that are being reported really are being strongly influenced by placebo or something like that. And so part of the reason in our upcoming study that we're looking at the immediate effects and trying to find markers of the state of microdosing is to see if there is any evidence of anything actually changing. But if you look at some of the lab-based studies that you mentioned, Elie, those studies are interesting because there haven't been too many of them. I think there's probably maybe 4 well placebo-controlled lab-based studies where experimenters have given participants a substance and then looked to see on various measures if there are any differences. And one thing that's interesting about those studies is that they don't find big changes in psychological kinds of effects, subjective effects, which does support the idea that, those sorts of effects might be driven by placebo. But they are reporting some findings that are curious. So they're things that maybe you wouldn't expect with microdosing. One study reported that there was a change in time perception. Another study reported that there was changes in the way that people perceive pain following microdosing. And then there's a couple of studies that have looked at neuroimaging and are showing some indications of changes in neural networks and brain activities following microdosing. So those sorts of findings suggest to me, at least, that there is something going on, but it seems like what is going on maybe doesn't match up with the popular culture story of all of the things that microdosing can improve and can do.
Yeah, and I know, David, in your review, you talked a little bit about the fact that many studies have found as a side effect anxiety as something that people report when they microdose, and you raised this idea that actually the anxiety might be because people are more in tune with their emotions and their memories, and it's coming to the fore, and it's making you anxious. But if you combine that with psychotherapy, perhaps it could actually be therapeutic. And so it fits maybe some of those things that you're mentioning, time perception, again, in isolation in a healthy individual. I don't really know what to make of that, but in the totality of mental health care, maybe those actually are very valuable responses. Any thoughts?
Yeah. I think it's possible. I mean, it's this unusual situation with microdosing because this is a sort of echoing what David said before. There is this intense public interest, and people are really curious and really interested in the field, which is great, and it's really exciting. But that interest is really outpacing the science and what we know, and it is hard to conclusively answer a lot of these questions. Like, science really does happen incrementally and slowly. There are these challenges around microdosing that David listed before around, unlike a high dose study, people have to do this many times, which means if you want to do a really well-controlled study, people either have to come in all the time or you have to give them substances, which is really problematic and tricky to get approval for. And so we can only sort of answer these questions one little bit at a time. And it seems like what we are going to eventually build up in our sort of castle of microdosing knowledge is going to be maybe a story that's not what people expect that it's going to be.
Yeah. For anyone watching out there, I'll just say we're going to start moving to some of your questions in a bit. So please put those into the question box and start to populate that. But I mean, one of the questions there is talking about the fact that, when you were saying that microdosing is 1/10 of a recreational psychedelic dose, I mean, some people take a gram of mushrooms, some people take 3 or more. So there's variability there. And it does seem interesting to me that when you look at the macrodosing world, a lot of that research has been done with pharmaceutical grade, either synthetic psilocybin or I'm not, Rob, maybe you can speak to this, or I'm not sure if it's purified or synthetic, but more to the point, it's a defined substance. Whereas all this microdosing research, it sounds like it's happening with whatever people get their hands on, and we're probably totally mixing up our conversations between different LSD and psilocybin, different compounds. And so I wonder, Rob, where you see the role of a company like Mydecine, maybe you'll kill me for mentioning your competitors, MindMed. Have they funded placebo-controlled trials with of microdosing with true defined formulations, and does that start to? Do we see anything interesting from that kind of research? Maybe just talk about that.
To just sort of jump into the questions there. So the first, simple answer is absolutely all the clinical research that's been done has been done with synthesized psilocybin, just a pure compound with a known, very specific dosing. And that is starkly different than what people are self-reporting with microdosing, because it is very much a I want to say, kitchen chemistry sort of dosing thing where they're providing themselves with a street drug or a product of nature, and then they're trying to arbitrarily dose that, but it's not absolutely, put through a pharmaceutical production facility where you're dosing. And so that's something where I do think companies like ourselves can really, stand and provide a lot of value to this question, because we are, employing a drug development pipeline that's really addressed to provide clinical grade, therapeutics for exactly these questions. And so I think that alone does really provide a additional, bump in the validity of this research as that gets, as that clinical supply gets overlapped with pointed clinical studies. I mean, these are the next steps beyond what we were just talking about, where we've had very good survey data that's already been taken. We've been moving into more looking at mechanisms and nuances within that survey data, and that's going to be really what paints the regulatory details that allow those next steps into pointed work. And definitely, as far as the questions for formulations, I think that's something that's completely open. And I think the state of the research right now is so much around taking the product of nature, the original form of these substances, or the original street drug compound in the case of LSD, and just understanding the potential there. There's a lot of questions as far as how that potential can then be best leveraged for use in therapy. And as a company, that's always something that I always try to strive to focus our research behind, is can we take this potential and then provide extra control to therapists, provide extra safety to the patients, et cetera, because those, I think, are the things that are very much addressable with formulation, like you mentioned.
Yeah. Are there lessons that you can draw, or parallels you can draw from the medicinal cannabis world, especially, in the sort of pharmaceutical line of things, where people have been, obviously there's a lot of stuff anecdotal and some basic research around flower, but there has been a whole kind of pharmaceutical pipeline as well. And clearly this field is years behind that. So are there, I don't know, are there things that they did right and wrong that we can draw on to try to accelerate this line of research?
Absolutely. And so there's, I think the two of them are most comparable at their starting state, where you have a large body of anecdotal knowledge around a natural substance, and the fact that there's, a growing amount of the population that's seen benefit from that personally. That's the kind of starting point that I see as similar there. The thing that's different, though, is that cannabis, as it was moved forward, is really kind of both a recreational and a pharmaceutical type, category, whereas what we're looking at here is, basically squarely a pharmaceutical type one, as far as the uses that we're looking at. But the theme of understanding the pharmacology of these products of nature, using things like molecular biology, understanding the specific receptors, like David mentioned, the specificity within the serotonin receptor system is very much relevant. And so using those same sorts of techniques that have been applied ad nauseum in the pharmaceutical space, and then have lent themselves more recently into the cannabis space to, really drive some of the really medically focused research there. Out of places like, Technion in Israel, there's some really strong research in that category. But that flavor of what's happened in the cannabis space, I think, is repeatable, and you can take best practices from the experimental design, et cetera, and apply it into this space. But the compounds, the nature of their use, and the best sort of medicinal value, I think, are starkly different at a certain point.
Well, what are the biggest, another comparable thing is that these molecules fall into kind of a regulatory and legal landscape of their own. I would think intuitively almost the psychedelics are harder to work with at this point, given the move towards legalization of marijuana in so many places.
Paradoxically, my experience in working in both industries is that it's actually the exact opposite of that. And it's because all the work that we're doing, and the vast majority, everything that we've been talking about on this call, and the vast majority of the industry is doing, is pharmaceutically focused. And so there's a very well-wrote, pathway of getting pharmaceutical drugs approved, researching them, et cetera. And so that actually, compared to the cannabis space, where there's a patchwork of different legal jurisdictions involved at best, is actually more straightforward. It's much more analogous to a regular pharmaceutical drug development business.
Okay. Has anyone ever, I mean, you can't do this blinded, but done like a rigorous comparison of macrodosing versus microdosing for the treatment of, say, depression, or at least some of the cognitive and physiological parameters that one would be interested in to really get at, not only microdosing, obviously there's benefits if you don't want to alter your your state of mind, you want to be kind of not hallucinating. But I mean, beyond that, are there clear ways where we can say where one might be more advantageous from a medical perspective than the other?
You're asking whether there's any evidence that
I'm just I'm taking directly comparing macrodosing for depression versus microdosing for depression.
I think that would be a complicated trial to do, actually, because people are microdosing 2 to 3 times a week over several weeks, and the full dose. So you need to make that work. And I mean, would you then? It's difficult to compare directly. I guess there is data coming out with small doses from the high dose trials when you are using a small dose as a kind of non-active control condition, because in a way you blind people better in a full dose study by saying that you will get psilocybin in each occasion. It could be very small doses, or it could be very high doses, so to keep people a bit in the blind in terms of the doses. So therefore there's an advantage, instead of giving them an inactive placebo in such studies, giving them a very low dose, because then they know they will get something, and that helps with the blinding, the placebo method. However, that will then produce data of does a dose and microdosing range do anything. I guess it won't if you don't have an inactive as well, but there do seem to be improvement from the very low doses in some of this work. And I guess the big Compass multi-center trials, they're also using different doses, including somewhere in the microdosing range. So we'll see whether there are improvements from there. But then you don't know, because then they don't have an inactive placebo. So I guess you should test that as well, by having all the conditions there.
There is kind of a building group of evidence around, if you look, like David's saying, at the doses that are actually provided in the clinical studies, there's definitely evidence that the low doses are having an effect, sort of linear fashion or somewhat linear fashion compared to the higher doses, in a dose escalation sort of fashion. So that's definitely promising in that avenue, but more data needs to come in, absolutely.
Yeah, and I think also, even if there's something there in those studies, that's a single dose, and we don't know how that would be compared to an inactive one. So but there's a lot of things that we can say, oh, that's interesting, but we probably also shouldn't be too becoming blind ourselves from focusing and picking out small little findings, because I would say, as I didn't mention when you spoke earlier about the acute dose studies with single dose, it is quite remarkable what they are not showing. But I think if you really want to stay optimistic, you can choose the small positive findings, which typically are not on the most significant, important things tested in the trials, and would be most interesting for further development for instance, treatment of depression or anxiety. I also want to mention one little thing, if that's okay, that in this self-blinding study that we are publishing now, that the expectation, we were actually assessing whether people were breaking the blind or not, and we can see that there's a dose-response relationship in breaking blind. So because there's a range of doses people are using, as you also were mentioning, Elie, in microdosing, they could use a tiny microdose, or they could use a quite large microdose. So there'll be a range, and when you correlate that to their ability to break the blind, to tell whether they were taking microdoses or placebo, then there's a relationship. So that means that if you get up a little bit in the high range in microdosing, they will be able to guess correctly that they have broken the blind, which is very similar to SSRI trials, where people actually also know what group they're in. Maybe because of side effects, they can also often break the blind, which potentially is a bit of a problem. and then if there's a dose relationship, and it's only the very, very low doses where you can really blind people and they don't show anything, then it's a bit of a problem. We also, in that trial, looked at a sub-sample of people who did score above into the clinical depression, in typically mild but also moderate range of depression. And we wanted to check whether there was any effects on depressive symptoms beyond placebo there, but it was the same as for the rest of the group. So that also doesn't point towards that it would be beneficial for depression. Unfortunately, of course, trials might be conducted.
All right, let's go to some questions that we're getting from viewers. One is about the entourage effect. So this is this idea that, I think it's used a lot in the cannabis world, that all the cannabinoids and terpenes together have an additive or synergistic effect that you don't get if you're just, say, vaping pure THC or something like that. And so is anyone exploring kind of if there's a difference there between kind of pure psilocybin as compared to consuming raw mushrooms, or everything is, I mean, maybe that's like too advanced a question because all the users in most of these studies are bringing their own drugs to bear. But can someone address whether there is any evidence for an entourage effect in?
Yeah, I'm happy to address that, absolutely. And so it's an interesting question, and certainly one thing that needs to be remembered is that the clinical studies that we were just discussing, those are all done with purified or synthetic compound. And so there's no entourage effect present there because it's only one compound. The opposite side of the coin, though, is that if you look at traditional use of psychedelic mushrooms, psilocybin producing mushrooms across a number of different cultures, especially when they have access to numerous species, they self-describe their use, their value, their spirituality, and their practice very, very differently. This suggests that they actually have different effects. And if you look at the reported chemistry that's produced in these mushrooms, they're making a number of minor tryptamines similar to minor cannabinoids. So they look very much like psilocybin, but they're specifically not. They have slightly different pharmacology. And then also recently, they've been shown to produce monoamine oxidase inhibitors, which are a category of drug compounds that actually make you more sensitive to the effects of psilocybin and other hallucinogenic drugs. And so if you take that as kind of a summation and assume that in the 200 plus species that actually produce psilocybin in the fungal kingdom, that you have variability there, I mean, this is the building blocks for having an entourage effect. But the idea of whether it's useful in a medical setting and can be leveraged for benefit in a medical setting is still a very much open question. So.
Okay.
Really, really interesting one, though, but complex, like you're saying, it's more complex than looking at a single molecule.
A question here for David and Vince. So this comes from Nicole, a student in Arizona, who asks for trainees who are interested in getting into psychedelic research. She asks whether you guys have any advice on finding a program that will support this line of research or a particular background, whether it's in neuroscience or psychology or what have you, that kind of best lends itself to getting into this field of research?
I guess so. The person asking is in the US. Vince and I are not there. So so in terms of where to be trained, that's a tricky.
She can travel for her PhD. I did.
Yeah, true. Oh, yeah, absolutely. I mean, I would choose what the person likes the most in terms of fields, because so many fields are relevant and interesting to be involved in research into psychedelics. So I think it could be. I mean, if you are medical, then you have an advantage in terms of direct access to clinical population that you work with. So you will be sort of dealing clinically with it, and you'll be allowed to administer the drugs in studies and so on, which you won't if you're not medical. But I mean, anthropologists, chemists, psychologists, nurses, everybody can be involved in research. There's no sort of proper formal education yet, but there are different. What is the one in California with IC? What is it called now? In
Center for Integrated Studies.
Yeah, I'm not sure, but there is a training program. I think it's costs some money. It's private, and I think you need to attend. Maybe not anymore because of COVID. Maybe they have done it all online, which would be quite smart. Yeah, and then I think MAPS are also training people. But if you are very early in your career, then I guess it's smart to have a clinical background in order to work clinically on the trials before that's relevant. But over the next few years, there will be more and more, I think, educational systems built into other programs, maybe into neuroscience and therapeutic therapy studies.
Is there something like an academic society that holds annual meetings or anything like that where it's a small-ish world where there's surely, there's hubs of this kind of research at Imperial, with yourself and David Nutt and others, Maastricht for sure, in this, Toronto, I think has, and you mentioned a couple of places in the States, but where would be a good place for someone like Nicole, back when we start meeting in person again, to try to meet all you guys? Or is it still too fringe a field?
No. I mean, go on, Vince. Sorry.
I was just going to say that there are psychedelic science conferences, often put on by MAPS. That's a good place to meet psychedelic researchers. Also, more and more in the last couple of years at conferences that aren't specifically psychedelics focused, there's more and more of this research occurring. So general cognitive science, consciousness type academic conferences certainly have psychedelic streams that would be good places to meet people. I just wanted to sort of add to what David was saying in terms of advice, is that if someone is, I think, relatively new in their educational career and wanting to get into psychedelic science, you don't necessarily need to start off with psychedelics as your specialty. In fact, it might be more beneficial to really try and build up some expertise or to build up some knowledge or background in some other field, whether that is clinical work or anthropology or cognitive psychology.
Or hypnosis.
Pardon?
Or hypnosis?
Hypnosis, yes. I did my PhD on hypnosis and altered states in general, which is maybe a little bit on the more unusual of pathways into it. But I think just having a really good skill set in some aspect of science and psychology can be a really beneficial way of then leveraging into this kind of work.
Yeah.
I think Vince is hitting the nail on the head there. I think, the majority of people working in the field have taken very sort of diverse pathways to get here. And it's really the ability to, leverage skills that are applicable to the field, but maybe were developed in a context that are really, aside, my own personal background is much more in developmental genetics, bioinformatics, that sort of thing. But as you, gain experience over time, it's something that you can definitely tailor that level of expertise, whether it's in pharmacology, whether it's in clinical therapy, or kind of anything in between. So I think it's a great time to be getting into the field. Absolutely.
I just want to mention, I think that what Vince was saying, that just strengthening through studying and maybe do studies in non-psychedelic field could, I think, is a nice way of preparing for later entrance into the psychedelic field, because you can, of course, try to seek towards some of the groups that are conducting psychedelic research. But the thing is, I would imagine that it's the same for other centers like ours in London at Imperial, that we actually get quite overwhelmed by the crazy interest. And I do understand this, people are enthusiastic. I mean, we are ourselves, and we are lucky to already work in the field, but it's horrible to disappoint quite a lot of people who want to get in. But I guess it's hyped not only out in the real world microdosing, but also the science, because it is exciting. But I think I would say where we are getting very interested in getting people in is typically when people already have some expertise in a method that could be applied to some of these studies. When people come with a method and want to address things, that's another way in. But then it's at a more senior point when you have already been doing other kind of research. But that's a way in if none of the groups can sort of have you in at this stage because they might be overwhelmed. But you could also be lucky that your CV is just amazing and or they just need somebody in the team. So you just have to try to seek towards the groups you're interested in.
Yeah. I'm going to use a question here to actually inject one of my own to bring back too. So we've been talking a lot again about microdosing in general. I'm curious, and a lot of the research has been on either LSD or psilocybin. But of course, you could start talking about ibogaine or ayahuasca or other things. And so I'm wondering if someone could speak to whether there are known differences when it comes to microdosing these various molecules. And then there's a question here from William who asks about some of the ethics, especially, maybe for Rob, about companies moving into this space, given that some of these molecules have important roles in indigenous cultures and just around the cultural sensitivities around commercializing some of these molecules, or even with the medical research, questioning some of those traditional practices. And I'm wondering, so it's kind of a two-parter, is one's looking at all the different compounds and whether there's differences, and then maybe some of these other issues as we move into turning what has been a practice for centuries, if not millennia, into essentially, Western medicine and a business.
Definitely. So I'll answer the first one, then go into the second one, because it's kind of two nested questions. So in the first one, yes, these substances are different, but our level of research right now is really focused on their activity at one receptor site, the serotonin 2A site, at least primarily. And so as we continue to do this research, there will be a lot of nuances, and I think probably best matching of certain substances, certain dosages with certain indications. But there's an interesting, sort of corollary with that, is that if you look at the safety profile across those, psilocybin is probably arguably the safest substance in that spectrum because of just sort of de facto, popular use and knowing the rates of hospitalization associated with, knowing the side effects that are potential. Certain substances that you just listed have other side effects that can be, if you were thinking of all patients possible, they would be excluded, if they had a heart condition, for instance, this sort of thing. So that's really how we look at that, is that, yes, that's a very interesting question to continue looking at, but at the moment we really are, thinking that psilocybin is a safe choice to be, looking at within that spectrum. And so that's a lot of why we've chosen psilocybin, because of the promise and safety aspect. The second part, the idea of how do we respect the prior use of these substances? How do we interact with the cultural use and value here? And the way I see it is it's really complementary and not the same at all. I mean, we're really talking about taking value that's been highlighted across a lot of human history and then understanding it completely in a medical lens. We're absolutely not looking to displace spiritual use, displace, these sorts of, traditional use practices. And I think, honestly, we talk a lot with different therapists and PIs in our clinical trials, and this is an ongoing long-term question. There's a lot of knowledge of how to use these substances in a shamanistic setting, but not every person in the world has a cultural background of, growing up in a society where shamanistic practice is really inherent. And so, one of the things that's kind of seen as a possible, mature solution here is to really train therapists and the people that will be, employing these pharmaceuticals, because they'll be more nuanced than what we're talking about as, these products of nature, and have them trained by an even-handed grouping of, traditional practitioners, modern, medical scientists, and then therapists as well, and, have a really, sort of holistic understanding of the history, and utility there. So that's really my best answer there, is that we're not looking to be competitive there, but we're looking to be able to, take the best value and provide it to as many patients as possible. And that's partly accelerated by, business practices and, the clinical pipeline, et cetera. So that's the full-winded answer there.
Okay. Question here from Monty asks, we're talking a lot about the use of microdosing for major treatment-resistant depression. But Monty asks whether there's any evidence for other. He asked specifically about kind of milder levels of depression, like dysthymia. I'm wondering about other things that have come up, like PTSD or other anxiety disorders. Is there any sense of where microdosing could help for other mental illnesses?
And actually, I can also think, is that full dose or microdosing or both? Because in full doses, it is being explored in major depression. It typically is targeting people with a significant, severity of depression. So very mild depression is typically not studied in these clinical studies because you. So in our study they were moderate to severe and resistant. The next study that we will come out with that has been completed was just in major depression, so not treatment resistant. So in a sense, potentially a bit less severe, but nobody has, as far as I know, done dysthymia.
Well, what about even in these like healthy individual studies? I assume a lot of people who are choosing to microdose, even if they haven't gotten an official diagnosis from a GP or a psychiatrist. Yeah, do you survey people, and do they almost self-diagnosed with this, persistent low-level depression or anything like that, and do you see differences?
Yeah, so I think I mentioned earlier that in the self-blinding trial we had a baseline these scales that are well validated and used for assessing severity of depression. We had people scoring over the threshold of depression, so into the mild spectrum. There was a significant amount among these healthy people that had sort of low, mild depressive symptoms. So we could just look at that group within our trial. So as a subsample, look at sort of depressed patients, even though they are not recruited and clinically diagnosed necessarily, but scoring above the threshold. And there were no effects beyond placebo in that group either. So as I mentioned, it does not look like, from what we know now, that it might work very well for depression. But there hasn't been doctored the proper blinded trials looking into it. But from our data, it doesn't look like it separates from placebo.
Okay. And we've got another question here from Keelan asking, I think this one's for you too, David, that about the risk of valvular heart disease by chronic microdosing of these serotonin receptor agonists. Now, you raise that as a concern, but is that particular side effect being systematically studied in any way to further delve into that risk?
No, and I think the simplest way would be in an animal model in rodents, potentially to test it, because it would be a bit tricky in humans. There might be people doing some echocardiography on patients to see whether or scanning to see the thickness of the heart valves, but nothing has come out yet, and I think that will take a while before that comes in humans. So the concern is not massive, but it needs to be tested further for sure. If it's
related to that, an interesting point with microdosing is it's not really well defined if somebody is, say, mildly depressed, or they're self-medicating with microdosing for a specific reason. It's not clear whether that's best to do for a month or 3 months and then try to wean yourself off and hope that that's been curative, or whether that's really the best practice is a maintenance medication for an indefinite amount of time. And both of those would change the perceived cardiovascular risk because of the receptor binding that David was mentioning. So.
Well, and that's a larger issue in all of psychiatry, with conventional SSRIs, how to wean, how to do it safely, when to do it. I mean, these ultimately, because those never get studied in clinical trials that are by definition, only a month or 2 or 3 long. So, but David, also you raised the issue. We hadn't really talked about this much. I'm wondering if you or Vince can talk about the preclinical space. I mean, are there good microdosing studies in rats or in any larger animals, and is useful information that can be drawn from laboratory studies like that?
I think
I'm not really across the preclinical work.
No, I'm not either, and in a way, this is going to sound horrible and arrogant. I almost don't care. We need to see whether it has clinical effects beyond placebo. Full stop. For me as a clinician to be interested. So I think that's the most important evidence. Of course, you can do. Normally, you do a lot of preclinical work in animal models, but that's when you start from scratch with the molecule. You need to see. But here we start a bit differently. We start with well-known substances. We already know a lot about them in full dose. But I think there's very interesting data coming from preclinical that just makes us even more interested and fascinated by the compounds. For instance, that there's evidence that psychedelics can induce some flexibility, plasticity in the brain. And that kind of work is very interesting. And we are, by the way, also going to test in the living humans' brains whether there is plasticity both with EEG paradigm and also with PET. The PET one is only for a ketamine study though, but hopefully future also with psychedelics. But some of that preclinical work I'm interested in, whether you have a model of, you can also have sort of depression model, psychosis model and so on in rodents, and then you can also test them there. But I was almost going to say, why bother in this field when we already are giving them and people are using them, millions of people are using psychedelics. So I think in this field, I think it's the human work that's more interesting in the next coming years.
Well, although I guess you could do some much more controlled studies. Like we've been talking a lot about the variability in certain things, whether it's the dose or the schedule, and because of all the ethics and logistical challenges of running the human studies, if you're interested in whether the optimal thing is 1/10 or 1/20 of a recreational dose or 1 day on, 1 day off, whatever, like.
Yeah, but I think that's very important to find out. But in humans, I mean, I don't think that's as interesting in a rat to see whether this little microdose does better than the other. That's very interesting. But we already using these compounds and seeing them and testing them in humans. So it's all down to whether it. Not that we don't care about rodents and how they feel, and if we can reveal that, help their depression, but we need to test it, and it will be, and it's already ongoing. There are people running trials. It will come in the future. I think you talked about MindMed. I think, isn't it MindMed who is going to run an LSD microdosing trial for ADHD? That's a bit interesting because that's one of the very few things we did not at all look at in our self-blinding trial. That's very interesting. I mean, what if that worked? That would be great. But I think it needs to come from human studies. And
Well, so one of the reasons that we're really interested in the biomarkers that Vince was mentioning with stress and with neurological function, because if you're thinking that there's a sub-perceptual effect that's really positive, and we want to understand the actual best dosage or the best timing for those dosages, without some sort of really, finite measure, it's very, very hard to do that. And so that's, undercurrent with that side of the research is that it should enable exactly the sorts of questions.
Okay, yeah. And I think even in that ADHD study, they're also looking at immune markers and things like that. So maybe that's the way to kind of get at all these uncertainties is to make the most use of biomarkers and really analyzing blood and do the brain imaging and get kind of the maximum amount of data out of the people who are.
Absolutely. I mean, if you think of any other field of medicine, they are always striving to have the best description of the condition that they're treating. And if you make a comparison, I always like to make to cardiology, in the mental health space, it's effectively like we don't have a cholesterol test. How would you ever prescribe cholesterol medication in a heart disease context if you didn't know what the cholesterol levels were? And so that's that type of test, and, knowledge over time is very much not at the same state in the mental health space.
Yeah, is there any sense that in the same way that a lot more venture capital or even government support has poured into medical cannabis space, like are we at an inflection point? Like if we had this conversation in 2 years, would it just be totally different and there'd be way more studies and we'd know more? I'm just curious, kind of in the last few minutes we have here, if someone can kind of give a forward-looking wrap up.
Yeah, I think this definitely is an inflection point, and I think the research that we're describing here, both on the micro and the macro dose, as well as the public interest that's really, really strong, is kind of the perfect storm, if you will, that's really pushing the utility of these treatments forward, and will prove whether they're useful as prescription treatments at scale or not, and that's really exciting. I think partly that would not be happening if we didn't also have matched increasing ability to look at these things on a mechanistic level. There's kind of all the pieces are fitting together exactly at the right time to answer the questions technically. People want to have these questions answered, and then there's a huge need. These are all very much underserved, treated conditions, and by and large, and so the promise of having better, more efficacious treatments is just huge.
David, I mean, where's your team going? With it now that you've wrapped up that study, kind of what do the next couple of years hold for you guys?
So although we are concluding from that paper, from the results, that it doesn't seem to be doing something on the key parameters, really, because of the weakness of the study, we are trying to close the gaps by basically do another study, see if we find the same, and we'll do that lab-based, so people, semi-lab-based, they'll visit the lab before and after. So instead of being a global trial where people can participate from all over the world, it'll be from people living in the London area who are, again, planning to self-blind with their own drug. But then if we are trying to figure out a way to measure the drug, so we see exactly what it is and the dose. And then we also do some neuroscience, including a plasticity paradigm with EEG, and also some BDNF measures of related to plasticity and other things we're going to do in that trial. So we are basically sort of fine-tuning the what we did in the first and trying to advance it further. And also, if that replicates the non-separation from placebo, then that's another nail in the coffin, as I see it, for microdosing. And I think all this thing about focusing on the mechanisms and blah blah is all very interesting, but if it doesn't work clinically, then I will at some point probably maybe lose interest as a clinician, because then it's not as interesting whether it increases this or changes that if it does not work beyond placebo, even in healthy people, for increase in well-being or creativity and all these things, which it doesn't look like it does in our trial.
And Vince, are you? I don't know if we have people from the Sydney area who are watching, but I mean, are you looking for participants, and kind of where's your work going in the first year?
Yeah, absolutely. We're hoping to start recruiting probably towards the end of February, and so if there is anyone in the Sydney area that is microdosing and is interested to take part, we'd love to hear from you. You can just email me through my academic profile, or I've also set up an address which is just sydneymdstudy@gmail.com. I'd love to hear from anyone who would be keen to take part.
And Rob, when do you think that a company like Mydecine will, I mean, the long-term vision, as I understand it, is not to just study people who bring their own drugs to the lab. So when do you think we might see kind of clinical trials involving, Mydecine formulations?
Yes, I think really the context that'll happen in is our PTSD focused work is really agnostic to the dosage level and the treatment form. And what we're doing there is striving for a series of investigator-led trials so that the investigators themselves, the PIs on the work, can really make those choices. And so in the spectrum of 5+ trials that are announced and then announceable in the near future, in that trajectory, I would absolutely, anticipate that we'll be addressing exactly this question of, looking at matching of therapy with exactly the correct dosage, and mixing and matching of macro and micro doses alone, et cetera. And I think that's kind of going to be where some of the rubber meets the road with this, is taking, iterative best case therapy practice that are the current gold standard, and then really, looking at different courses of dosage across the same treatment. And so that's really, it's a longer-winded answer, but that's where I see it fitting in with the company's research.
All right. Well, you guys have convinced me. I'm excited by at least, what the research has in store. A lot of questions, a lot of early promise, but definitely need to see the field mature, and be interested to see David's paper when it comes out. Shoot me a preprint maybe, and Vince, good luck with the recruitment, and Rob, all fascinating stuff you're doing too. And so thank you to all three of you. Thank you to everyone who's been watching and tuning in and asking interesting questions. And I think any of us, our email addresses are available if you do a bit of clever Googling, and you could follow up, and hope to continue the discussion sometime soon. So thank you again.
And I just want to say thanks again to Mydecine. Thanks for coming on, guys. And just to let the audience know that if you do feel like sticking around, we have an informal networking thing going until 4. So if you go back in, you can find people and do some sort of speed matching. So thanks again for coming on, guys. We appreciate it. It was a very insightful conversation, and we look forward to the next one.